So thank you, Elizabeth, for your kind introduction. And thank you to the audience for joining the webinar today. I'm going to introduce you to the topic of the evolution of contract research organisations to contract innovation and partnership organisations. So on the agenda today, I'm going to be talking to you about the evolution of contract research organisations to contract innovation organisations. And I'm going to discuss this then in the context of Eurofins Discovery and our Discovery One Integrated Drug Discovery Partnership Platform. I'll go through medicinal chemistry, business models within Eurofins, and then share with you a case history of a complex drug discovery project that's initiated from a really challenging start point. So the pharmaceutical industry is facing ever-increasing pressure, not just to fill the pipeline, but to deliver value to the payers as well as the shareholders. Over the last decade, one of the ways that companies have sought to reduce costs during the research and development phase is through externalization of research to contract research organizations. Outsourcing research that would previously have been carried out in-house allows capital efficiency by moving fixed costs into variable costs, and also capital elasticity with the ability to adjust spending by portfolio needs and data. Outsourcing can also be used to address capacity shortfall, an internal expertise or capability gap, and allows companies to focus on their core expertise while externalizing other activities. Strategies towards outsourcing vary between organizations. Some companies prefer to use a wide variety of different suppliers, seeking the best specialists for the different components of the drug discovery process. Others prefer to work with providers of integrated drug discovery, where a multidisciplinary project team's drug discovery experience is brought to bear on a project from within a single CRO. Historically, ownership of project strategy and direction was wholly within the pharma bio, with CROs tasked with transactional services and execution of projects only. This meant many different interfaces with different suppliers for the pharma bio. Things have moved on, and pharma bio companies are entrusting more and more of projects to their partners, with transactional relationships providing services evolving towards partnership relationships and providing integrated solutions. Rather than the multiple different interfaces, projects are run in an integrated fashion under a single MSA and with an integrated project manager. Typically, there is a project team on both sides who operate in a peer-to-peer manner on a scientific basis, with overall control of project strategy by the pharma bio. CROs are moving towards providing the strategic plan as well as executing on the plan, and have authority to make decisions up to a certain point. Moving towards the future, innovation as well as solutions are coming out of the CROs, and blurring of the edges is occurring as to where the project initiates. Business models incorporating risk-reward sharing are becoming more common, and levels of trust in CRO partners are rising, driven by high transparency and visibility. A CRO that puts good communication and high visibility in all aspects of its services has a good opportunity to build trust and long-lasting partnerships with clients. Decisive governance through joint steering committees with clear accountability are required to run these partnership projects, and high accountability now is lying within the CRO. So in previous times, Eurofins was one such CRO working in transactional services. So, for example, running selectivity screening or carrying out single projects for customers. Things have moved on, and Eurofins Discovery is certainly in the realms of working in partnership projects of today and already also of those partnership projects of tomorrow. Moving on to discuss Eurofins. So Eurofins Scientific is an established leader in life sciences. It has 4 key divisions: biopharmaceutical, clinical diagnostic, environmental and food, and over 400 labs and over 40,000 staff worldwide, with revenues of over 4 billion. Eurofins Discovery fits within the biopharmaceutical division of Eurofins Scientific. And Eurofins Biopharma Services division as a whole spans the whole target ID to product space in covering biopharmaceutical testing as well as product delivery. Eurofins Discovery sits in the early discovery space from target ID and validation up to preclinical candidate, and then works closely with our Eurofins CDMO and early development partners to enable a customer to remain within the Eurofins group from target all the way to IND and then beyond into the clinic and through to launch. Eurofins Discovery has acquired best-in-class companies in order to be able to offer integrated drug discovery services. In terms of the individual companies acquired by Eurofins Discovery and now operating under the Eurofins Discovery name, the chemistry companies were BioPharma and Celsia, who together offer synthetic and medicinal chemistry, integrated drug discovery, and project management. DiscoverX and the Merck Millipore, formerly Upstate and Chemicon, Discovery and Development Solutions, are 2 of our biology companies with deep expertise in a number of different target classes, and additionally have phenotypic capabilities. Syrup and Pan Labs are extremely well known for the safety capabilities and indeed support integrated drug discovery projects, not just through the safety expertise, but also through DMPK, in vivo, and ADME services. Eurofins Discovery are a global organisation with 600 plus passionate scientists, lowest lab turnover rates in the industry, and the longest successful in vitro track record stretching back over 40 years in the industry. Furthermore, we are committed to our clients with no competing internal drug discovery projects, and over the years have generated billions of data points for customer projects. In terms of our capabilities, we have chemistry, in vitro, phenotypic, ADME and toxicology, in vivo, and products. And within those divisions have scientific teams who work in collaboration to deliver preclinical candidates for our customers, stretching from the early discovery space through hit finding, hit to lead, and through into the lead optimization phase, as well as preclinical. In terms of the individual capability, project management is essential for integrated drug discovery projects, and we coordinate on behalf of our customers throughout the Eurofins network in order to bring all the capabilities required by the customer together to work for the best on their project. Within the chemistry space, we have library design and knowledge-based design, as well as computational capabilities and compound logistics. Our in vitro pharmacology group are extremely experienced, and in terms of capabilities, CRISPR is a new addition into the group, molecular biology and assay development capabilities, as well as cell line generation, high throughput screening, and primary pharmacology. Assays to support lead optimization, as well as selectivity panels, secondary pharmacology, and on through into preclinical safety panels. In phenotypic, we have human primary cells, biomarkers and iPSC capabilities, phenotypic screening, and through lead optimization can support projects through cellular pathways and biomarkers, human disease models and systems biology. ADME capabilities support both the early stages of projects as well as through conventional hit to lead and lead optimization stages. And we have in vivo capabilities which can be used at an early time point in a project through in vivo validation of the target. We also have animal model development capabilities and can carry out in vivo efficacy and PK/PD on multiple targets. In fact, over 250 animal models within the group. Our products can also be used to support any of our integrated drug discovery projects. The way we work with customers has evolved, as I say, from the more transactional single scope projects. These days, we're working more in partnership with customers, carrying out iterative projects, for example, carrying out screening on targets to support customers' lead optimization projects. In the integrated drug discovery space, then we combine the capabilities across the Eurofins group and align a project team under a single project manager in order to bring all those capabilities and drug hunting expertise from within Eurofins Discovery to bear on a customer's project. We also have a modular way of working with customers. So, for example, we also carry out integrated biology projects whereby, for example, a customer may be working with their own internal chemistry and so only require the integrated biology services from Eurofins. And in these cases, we will work closely on those projects, providing the customer with, for example, in vitro pharmacology, ADME, and in vivo projects. Now, we don't just work in a transactional manner with customers in these partnership projects. We advise on strategy. So, for example, helping customers design their screening cascades and advising on the medicinal chemistry strategy, for example. Integrated drug discovery offering is offered as a platform called Discovery One, integrating discovery excellence. Our value proposition is as your hand-in-hand partner in drug discovery, driving accelerated delivery of your preclinical candidate with integrated project management, deep scientific expertise, and broad in-house capabilities. Our platform goes across from target identification and validation all the way up to preclinical candidate and then beyond within the Eurofins group through our other Eurofins early development colleagues, and is underpinned by the project management, the drug discovery expertise, and the extensive resources and capabilities from within the Eurofins Discovery Group. So the chemistry, in vitro, safety and efficacy, ADME and tox, phenotypic, in vivo, and also the custom services. So how could Discovery One, our integrated drug discovery platform, help you? Well, we have over 2,500 robust and validated assays available and bring creativity to your assay development. So many of the assays that we offer through Eurofins Discovery are actually designed within Eurofins Discovery and indeed patented by Eurofins Discovery, and so offer clear benefit to customers when they're working with the people who actually design the assays in the first place. We're continuously seeking to bring in new technologies to benefit programs. We collaborate with you to help design your screening cascade, support you with medicinal chemistry strategy, design and synthesis, and bring innovative approaches to your project, as well as managing these complex multidisciplinary projects. In terms of our track record, our team has delivered over 50 candidates, over 15 in the clinic, and generated over 200 patents. So, of the selection of candidates showed here, across a range of different therapeutic areas, patents are owned by customers, though our scientists are named as inventors on the patents. All IP is assigned over to the customer. So in terms of these particular projects, there's one here in pain, phase 1, some very high-ranking publications in journals, including Nature. And the topic of the case history that I'm going to focus on today is this viral infection study on a complex protein-protein interaction target, where we delivered 2 preclinical candidates for HCV infection and generated 2 medicinal JMedChem publications as well. Now, talking about CROs moving from service providers to solution providers and then on to innovators, here is an example of where a customer came to Eurofins Discovery, and we approached that customer, Parkinson's UK, to provide integrated drug discovery services on targets that they had identified. Now, they gave us 3 targets to look into, and during the process of researching their targets and putting together a proposal, including our screening cascade design for those projects, our medicinal chemistry design for those projects, our scientists identified, simply out of the literature, an alternative target that they thought would be a useful target for the treatment of Parkinson's disease. So we delivered the proposals that Parkinson's UK had requested on their desired targets, and also inquired would they be interested in hearing about this alternative target that our scientists thought could be useful to them. Indeed, they were interested, and the outcome was that they preferred the target that we had identified than their original targets that they had asked us to write proposals on. And so we initiated a drug discovery project in January of 2018, supported by Crescit for virtual screening, on this project that had been identified by the Eurofins Discovery scientists. So this project, although I can't report the outcomes of it at this particular time, has met its 12-month milestones, and the project has progressed into its 2nd year. In terms of the medicinal and synthetic chemistry expertise within Eurofins discovery, which can be brought to bear on integrated drug discovery projects through our Discovery One platform, we have a team of experienced multidisciplinary drug hunters, with the medicinal chemistry design team having an average of 19 years experience, predominantly gained in big pharma. And this team works collaboratively with our multidisciplinary teams across Eurofins to support your projects and apply their extensive experience to drive your project forward. In terms of modality, our team have experience with fragments, small molecules, macrocycles, natural products, and small linear and cyclic peptides. In terms of the size of the team, this is a scalable team of over 100 chemists. And in terms of capabilities, we have, as outlined here, fragment screening, libraries, diversity libraries, library design, can carry out virtual screening, knowledge-based design, and consultancy and support for grant proposals. We also are extremely experienced in the evolution of screening hits, SAR assessment and compound exploration, screening cascade design, and support of project management across the group in terms of the coordination with pharmacology, ADME, and PK. In silico tools, computer-aided drug discovery, scale-up chemistry, and compound management and logistics, as well as IP generation, are all core capabilities within the team. In terms of our medicinal chemistry options, we have various different business models which customers can tap into at various different levels of cost. So we offer a traditional medicinal chemistry team. This is a co-located team of medicinal chemists, all of whom see the data that's coming in across the whole project, analyze data, and input to the design of the next iteration, the next generation of compounds, and then synthesize them. On the other end of the scale, there is the pure synthesis capabilities within Eurofins. And this is where the customer carries out all the internal medicinal chemistry design, but wants to tap into the integrated services within Eurofins Discovery and carry out just the synthesis in terms of the chemistry. And we support this model as well with a project manager to coordinate the multidisciplinary integrated models. Now, we also have two flexible ways of interacting with both these types of team. So firstly, there is the designer and synthesis model. So this is where a shared medicinal chemistry designer works with a pure synthetic chemistry group to support customers' medicinal chemistry projects. So that is a single individual who can be shared with other companies to support the medicinal chemistry design. Our other model is the mixed model, and this is where, rather than having a single individual responsible for the team who has responsibilities to other customers, this is where we have a team of 3 or more dedicated co-located medicinal chemists, including the project leader, all of whom can analyze the data, and they design and synthesize compounds and can challenge the strategy. So there's ideas and input coming in from this medicinal chemistry team. However, this option brings with it much lower pricing than having the whole team as traditional medicinal chemists, because this small team, where you're getting input from multiple people, are supported by the synthetic chemistry group, thereby making the overall project more affordable. So all these different options are open to customers, and customers can pick and choose between these models, and Eurofins can support these different ways of working for customers. And we also have a starter model whereby you can try us out on a 3-month FT contract to see how we get on. Now, in terms of the biology capabilities at Eurofins, these are extremely extensive and in fact are the subject of a separate webinar on in vitro pharmacology, which is happening on November 2nd. So please come along and find out more about our in vitro pharmacology at this webinar. Just briefly then, as this is the topic of a different webinar, in terms of the number of assays we have within the Eurofins group, over 2,500 robust and validated assays, which means if a customer wants to use this assay on a project, it's there, ready and waiting, no need to carry out any assay development, and shortens the timelines for projects because the assay is already in place. We have diverse libraries, ensuring ample chemical diversity for your project, and a total capacity of 500,000 assay wells per day, which can accelerate hit finding timelines. Lead optimization is well supported within Eurofins Discovery with decades of assay development and assay troubleshooting expertise, ensuring high quality data to keep projects on track. Turnaround times can be as little as 24 hours, depending on the projects. Eurofins is well known for its comprehensive selectivity and safety panels, and hundreds of unique assays and assay technologies make Eurofins the only solution for many projects, and we're certainly the industry leader in safety pharmacology with several gold standard panels. In terms of target class kinases, we have comprehensive expertise and experience, 20 years of screening expertise with over 50 million data points screened, and over 440 unique human wild-type kinase targets are offered on orthogonal platforms. We also have over 100 cell-based assays to study a compound's functional effect on the cell. GPCRs as well, a key area of expertise, comprehensive technology covering 90% of the GPCRome with over 1,800 assays across 6 different assay formats. Ion channels as well, so comprehensive ion channel experience across multiple different target types, and in addition, all these alternative targets are available within the Eurofins in vitro pharmacology offerings. In terms of ADME-Tox, again, we are well set up to be able to support any ADME during hit to lead through preclinical candidate selection with a comprehensive ADME-Tox menu covering a Tier 1 and Tier 2 panels. Phenotypic biology as well, comprehensive experience used for translational research to de-risk compounds and programs for subsequent development. And we have multiple capabilities in human-based phenotypic screening with complex human primary co-cultures which more closely model human disease biology for phenotypic screening. In vivo safety and efficacy, Partner Lab Pharmacology Discovery Services has over 25 years of experience conducting small animal in vivo pharmacology. It's a rodent-only non-GLP vivarium with all the accreditations that are expected, and dedicated staff averaging over 10 years of experience in vivo pharmacology. In terms of the therapeutic models, we have over 250 standard in vivo models for 8 key therapeutic areas, so anti-infective, inflammation and allergy, CNS pain, oncology, metabolic, endocrine, respiratory, and GI, as well as models for safety tox and DMPK. I'm now going to switch gears slightly and tell you about a case history where Eurofins Discovery initiated a project and took this around to a number of companies who we thought may be interested in working with us to deliver a cyclophilin inhibitor for the treatment of HCV. More than one company were interested in working with us on this project, and we picked a world-leading antiviral company as our partner on this project. What Eurofins Discovery brought to this world leader was our internal biology and chemistry expertise on peptidyl-prolyl isomerase, known as PPI targets. So we had developed within Eurofins Discovery cyclophilin assays, both TR-FRET and biochemical functional assays, to support this project. Now, the cyclophilins are a family of peptidyl-prolyl isomerases, which are chaperones of protein folding. So they catalyze the cis-trans isomerization of peptidyl-prolyl bonds. Now, cyclosporin is a very well-known clinical compound that is an inhibitor of cyclophilin. And it had been demonstrated that cyclosporin itself and some non-immunosuppressive derivatives of cyclophilin were potent against HCV replicon activity. Now, indeed, when we initiated this project, there was already human clinical data supporting that a cyclophilin inhibitor would be useful in the treatment of HCV. However, from our knowledge of the compounds and of their progression, we were aware that these were unlikely to reach the market, which gave us and our partner an opportunity to identify a synthetic, non-cyclosporin-based cyclophilin inhibitor for the treatment of HCV. The cyclophilin as a target has been around for many years, but all of the clinical compounds were based on this cyclosporin scaffold. Our goal was to identify a non-cyclosporin cyclophilin inhibitor, and no small molecule inhibitors of cyclophilin, even now to date, have led to cell-active inhibitors with oral properties beyond our own project. When this project initiated, we were set out looking for conventional small molecule start points, and indeed carried out a pilot HTS and a fragment screen. However, were unable to identify any promising leads through this initiative. We then went back to the literature and found an alternative natural product which had been published by Novartis called cyclophilin, and this demonstrated potent cyclophilin inhibition and also HCV replicon activity. Furthermore, a semi-synthetic cyclophilin analog was active in the HCV replicon assay and non-immunosuppressive. So from this start point, our goal was to identify from this natural product start point a fully synthetic, orally bioavailable, non-immunosuppressive small molecule cyclophilin inhibitor with potent anti-HCV activity and low drug-drug interactions that could serve as part of an all-oral antiviral combination therapy. So Cyclophilin itself, Novartis had carried out a fair amount of structure activity relationships and medicinal chemistry on the scaffold itself. And they had also obtained a crystal structure of cyclophilin bound to cyclophilin, which showed key hydrogen bond interactions between the molecule and the target that were responsible for the potent binding. Furthermore, they had demonstrated that one could remove the spirocyclic side chain of cyclophilin and that the potency was found within this macrocyclic part of the molecule. They had also carried out a synthetic chemistry campaign to simplify the structure of this macrocycle and generated a number of fully synthetic analogues. However, all of these analogues were at least 100-fold less potent than the natural product start point. This work was published in JACS, and when we analysed the SAR, we could not really work out why they had lost all this activity, because multiple step changes had been taken between the natural product and these simplified derivatives. We decided to set out and make stepwise single point changes from the natural product to our simplified analogues in order to determine the minimum pharmacophore required for binding. One of our early analogues that we synthesized was compound 1, and this pleasingly showed potent cyclophilin binding activity as well as some activity in the HCV replicon assay. Here we had removed this ketone-containing side chain and also this spiroketal-containing side chain, which Novartis had demonstrated was not required for efficacy. However, importantly, what we had kept in the molecule was this metatyrosine, and all 4 of these chiral centers are around the right-hand side. And there is an intramolecular hydrogen bond that you can see in the crystal structure, which we realized was quite important for the potency of these early compounds in combination with this diene unit in the northern region. Moving forward, we rationalized that the other 2 chiral centers within the right-hand side were not really interacting with protein and had no intramolecular hydrogen bonds themselves. So we removed both of these chiral centers, and we were also cognizant that a diene is not a great moiety to have within a drug substance. They're commonly associated with light sensitivity, for one thing. And so we looked to replace the diene and introduce this styrene in the northern region. Now, this compound, we obtained a crystal structure bound to cyclophilin. And when we overlaid that with the structure of compound 1, which contains the diene and all 4 stereocenters, we were able to see that we'd had really quite a dramatic change in binding mode. So, for one thing, the metatyrosine, which was previously thought to be critical for binding through its H-bond interaction with histidine 126, has rotated and in fact undergone a hydrophobic collapse across the center of the macrocycle, making a pi stacking interaction between that metatyrosine and one of the protons on the styrene. Now, in the northern region, there's also been really quite a dramatic change in binding mode. So we've moved from edge-on and hydrophobic interaction to tight pi stacking with the pi face of the guanidinium group of arginine 55. Now, this change of binding mode turned out to be absolutely critical for the progression of the project and enabled us to identify potent analogues whilst being able to simplify the structure of the macrocycle still further. Much work carried out in the diene series gave much less flexibility to be able to simplify the structure. So really identifying this crystal structure early on in the project was super important for its later success. Now, as this metatyrosine was no longer binding to the target, we rationalized we could remove it, therefore simplifying the structure still further. And also, it's a non-commercial amino acid, so we changed that for a commercial one, so reducing the cost of the synthesis as well. And indeed, this compound 4 was also showed good potency in both binding assays and also in our replicon assay. And this early stage of this project has been published in the Journal of Medicinal Chemistry in 2017. The project then progressed and we took a look at the properties. The one HCV genotype which these compounds seem to be weakest on was 2A. So the compounds were very potent on 1A and 1B, but we really needed to improve this HCV 2A potency, and this is what I'm going to focus on in the rest of the presentation in terms of the potency. Now, we identified other properties that required improvement. So the compounds were not particularly stable in human or dog, and in fact were completely unstable in rodent, to the extent where we, in this series, didn't really look at rodent clearance because it was cleared instantly. Permeability in this early series looked good in terms of the Keiko A to B values, so over 10 here for permeability. But the log D was a little higher than we liked. And we wanted to see whether we could move away from this styrene scaffold, as there are issues with styrene as well as drug substances. So we formed bicyclic moieties at the top and introduced this nitrogen, so the isoquinoline, in order to bring the log D down and not to make the compounds too lipophilic. This was also an attempt to bring down clearance. This isoquinoline moiety was tolerated in terms of its potency and in fact improved the HCV2A replicon activity, and was not too detrimental either to the forward permeability. Now, earlier in the presentation I mentioned the presence of an intramolecular hydrogen bond between the O-methoxy on the side chain and the NH of the valine. Now, because we had introduced the strong pi stacking between arginine 55 of the target and this bicyclic moiety, we were able to remove the intramolecular hydrogen bond and maintain activity, whereas in the initial diene series, that was completely impossible. So we were able to remove that intramolecular hydrogen bond, and that did improve our clearance slightly, but noticeably it has greatly reduced our permeability. And this is because the removal of that O-methoxy has unmasked an additional hydrogen bond donor. What did make a dramatic difference to our microsomal stability was actually shrinking the ring size and moving from a 22-membered ring to a 21-membered ring. We gave a compound that was potent in our TR-FRET assay and reasonable potency in our 2A replicon, and whereas the permeability of this compound was not great. This actually translated into low oral bioavailability in both dog and cyno, and so therefore improving those properties was the focus for the medicinal chemistry design going forward. We looked at regaining permeability with two strategies. Firstly, we had introduced this isoquinoline group, so on the outside of the molecule, and we thought that moving that into the center of the macrocycle would bury that polar group somewhat and improve permeability. So we moved from compound 7, the isoquinoline, to this internal quinoline group, which maintained the positive human clearance properties and did improve the KCO2 forward permeability. We also had identified that these compounds had moved away from the cyclophilin target on this, what we call the eastern region here, and we felt that introducing additional moieties at the 14 position could fill that gap and bring the compounds closer to the target, and there was potential for a positive interaction with threonine 73. So we introduced gemdimethyl at the 14 position, and that has the added benefit, as well as improving potency threefold, of improving the forward KCO permeability as well, whilst having the log D still in a reasonable range. Now, what was actually a critical finding for this project was when we went to the effort of attempting to solve the rodent instability of these compounds. So we determined that the instability was through the action of esterases on this lactone moiety at this position. And we tried various efforts, including bulking up in terms of sterics around this position to try and prevent hydrolysis. However, these tactics either did not improve the hydrolysis issue or led to less potent compounds. And where we found success was actually maintaining a lactone in the moiety, but moving it to a different part of the molecule. So we did a lactone-lactam swap and moved the lactam from the 12 position over to the 1 position, and had the lactone on this eastern region where it's much more sterically hindered. Now, if we had a bis-lactam, then unfortunately the log D dropped and also we lost potency. But this compromised position of having a lactam-lactone, having moved the lactone, gave us a compound 10, which had excellent properties and really improved the rodent stability from being completely unstable in rodents to being really good in terms of stability, and also dramatically improved our oral bioavailability with excellent oral bioavailability both in rat and dog, thereby meeting our target product profile criteria of oral bioavailability of over 50% in 2 species. Now, the solubility of compound 9 was also a challenge and something that had led to the low oral bioavailability of compound 9. Now, the solubility of this lactone-lactam was increased, and this was certainly helpful towards the excellent oral bioavailability that we observed. So altering the environment of a hydrogen bond donor has had really quite a dramatic effect on the properties on this scaffold. What's interesting here as well is that if you look in the crystal structure, there's extremely good overlay of these two molecules when bound to the target. And in fact, you can't see the intramolecular hydrogen bond when the compound is bound. However, these molecules are acting as molecular chameleons, and we have good evidence from their NMR in different NMR solvents of different lipophilicities that these compounds form the intramolecular hydrogen bond when in different environments. And this is extremely helpful for crossing cell membranes. The final optimization to candidate focused on fixing an issue that came up during our scaling the compound up and profiling towards being a preclinical candidate. And unfortunately, the PXR activation profile of compound 10 was suboptimal. However, this compound was extremely close to being a preclinical candidate within its other properties, and therefore we stuck closely to that scaffold in order to make small changes and to deliver a preclinical candidate from this scaffold. Our work focused on the eastern region and delivered compound 11, which fitted all the criteria that had been set out to meet preclinical candidate selection, including good oral bioavailability across 2 species and predicted once-a-day dosing in man. So to summarize this project, we started from an extremely complex natural product and were able, using structure-based drug design, to simplify that structure to give an oral preclinical candidate. During the course of this project, we synthesized over 450 macrocycles and screened them. We carried out scalable and convergent syntheses of these compounds, filed multiple patents, and identified a compound with PK properties consistent with once-a-day dosing in man, oral bioavailability of greater than 50% in 2 species, and low drug-drug interaction potential. So our candidate has a much lower molecular weight than our start point, far fewer hydrogen bond donors, which is helpful to its favourable oral properties. We've dramatically decreased the complexity of the synthesis by moving from something with 17 chiral centres down to 4 in the eventual candidate, of which 2 of those were commercially available. The potency was equivalent to the start point, and the developability was far more. We've published now 2 papers on this topic, the first in 2017, and we're really delighted that our 2018 JMed Chem was selected as a featured article by the Journal of Medicinal Chemistry, and in fact had a viewpoint article written about it and published alongside. So to summarize our achievements on this project, by working with Eurofins Discovery 1 Integrated Drug Discovery Team, the partner was able to access cutting-edge drug discovery and medicinal chemistry tools and expertise to generate synthetically accessible small molecule analogues of the complex natural products, access challenging target class biology expertise not available in-house, to quickly initiate a high-risk research project without building a new in-house team, to identify and advance 2 promising anti-HCV drug candidates, to file 4 patents surrounding this novel mechanism, and to publish 2 journal articles. So just in summary, I think I've discussed with you today how CROs, including Eurofins Discovery, are evolving from being transactional service providers to companies that provide solutions but also provide innovation. By partnering with us, you tap into your 40 years of experience of providing high-quality drug discovery services and products, a reputation as a trusted service provider, expertise, comprehensive portfolio of services, flexibility, and integrated drug discovery project management. And thank you for listening.